Hello, my name is Monica Embers, and I'm going to speak today about persistent Lyme disease with some supportive evidence that we've found and some potential solutions. As many of you know, post-treatment Lyme disease is a significant problem, and many patients are successfully treated if they're treated early. But it is broadly accepted that there is a sizable number of patients who experience treatment failure and to continue to suffer long-term debilitating symptoms. And this publication from 2019 used an analysis to combine the cumulative number of patients who were suffering from post-treatment Lyme disease and found it to be in the United States close to 2 million in the year 2020. So this has probably increased over time. In terms of the clinical case definitions of post-treatment Lyme disease, there are a number of publications demonstrating this, and they are distinct from other manifestations such as fibromyalgia and chronic fatigue syndrome. However, the three primary symptoms associated with post-treatment Lyme disease are musculoskeletal pain, fatigue, and neurocognitive difficulties, which ironically is very similar to long COVID. So in terms of post-treatment Lyme disease, there are a couple of different possibilities in terms of the causation. The first is the induction of inflammatory responses by lingering dead spirochetes or antigens from the spirochetes. We know that the Borrelia peptidoglycan is very unique and that it on its own can induce inflammatory immune responses. The second is the continuation of active spirochetal infection, and this is what we study in our lab primarily. And then the third is irreversible sequelae from a previous active infection or the so-called autoimmune etiology. And most likely all three of these are acting in concert with post-treatment Lyme disease. So in terms of antibiotics themselves, there are a couple of considerations to think about. Doxycycline itself is a microbiostatic antibiotic. It doesn't kill the bacteria directly, but relies on immune clearance of non-growing bacteria. And we know that Borrelia burgdorferi evades the host immune response in many different ways, and persistence is the norm in immunocompetent hosts. We know that the bacteria can become dormant, and that those dormant spirochetes or slow-growing spirochetes are more tolerant of microbiostatic antibiotics such as doxycycline, and I'll discuss resistance versus tolerance. Borrelia burgdorferi is known to survive many months inside ticks without any nutrient replenishment, so dormancy is definitely part of their life cycle. And finally, Borrelia burgdorferi can be found deep in connective tissues and joints, so we have to think about how well those antibiotics penetrate the tissues. So the IDSA, or the Infectious Disease Society of America, has treatment guidelines, and I just want to focus here on patients with disseminated disease, and the recommended course of treatment is doxycycline or amoxicillin for 28 days. Whereas if you look at the ILADS, International Lyme and Associated Diseases guidelines, it's a little bit more flexible, and they recommend 4 to 6 weeks of doxycycline, amoxicillin, or cefuroxime, and also open it up to the discretion of the clinician in terms of retreatment. Saying that antibiotic retreatment when a chronic infection is judged to be a possible cause of ongoing manifestations, and the patient has an impaired quality of life. And we know that happens frequently with post-treatment Lyme disease. So in terms of the antibiotic efficacy against Borrelia burgdorferi, we've shown in studies that the development of persister, that the Borrelia develop persister cells, and we know that it's most commonly treated with microbiostatic antibiotics. And if you think about it, all the determinations of the minimum inhibitory concentration, that is the dose that we use to treat people, have been performed in vitro, so in a cell culture system. And we've done studies, which we haven't published yet, but they basically demonstrate that when the bacteria are host adapted, then they become even more tolerant of the antibiotic. So the MIC levels that we test in vitro really aren't accurate. We've also shown in non-human primates that the spirochetes persist after treatment of a disseminated infection. We don't know of any specific mechanisms of resistance for Borrelia burgdorferi. This is not an antibiotic resistant mechanism. We know that the tolerance to antibiotic is achieved with slow growth, that host adaptation contributes to tolerance, and there are studies that have deemed these bacteria as viable but non-culturable. And I'm going to talk about that as well. So this is basically a diagram showing the difference between resistance and tolerance. And so if you have an antibiotic resistant bacteria and you add the antibiotic, those bacteria will grow normally in the presence of the antibiotic. However, if you have bacteria that are tolerant, the bacteria will not die, but they will also not grow. So they'll basically stay in this dormant phenotype throughout the course of infection. Get my pointer here. There we go. So this is these are resistant mutants and these are tolerant bacteria. So back to the viable but non-culturable state. This is not unique to Borrelia, just like dormancy and persistence is not unique to Borrelia. It occurs in over 100 different bacterial species studied to date. There are 51 pathogenic VBNC bacteria in humans. What we know is that the membrane integrity stays intact, but there are structural changes that are aimed at increasing the surface to volume ratio. And we see this with the different forms of Borrelia. And there's really no great evidence to distinguish VBNC cells from persister cells. This has oftentimes been used interchangeably, but it's thought that there are different levels of dormancy. We know that they're alive, they're metabolically active and viable, and many of them can be resuscitated. So in terms of VBNC bacteria, there is a broad phylogenetic distribution. There are common characteristics, but there are also differences in the induction of the VBNC state and the conditions required for resuscitation. Some remain infectious after regrowth and some do not. So this was a Ymir Hodzik study and Stephen Barthold back a few years ago. And this is where the VBNC state was observed in mice. And we've shown this in primates as well. And what I love about this mouse study is that it took patience, right? So what they did was they treated mice with antibiotics after a disseminated infection, and then they waited for different periods of time. And after 8 months, there were still very, very low levels of spirochetes. But when they waited 12 months, a year after the end of treatment, the levels of spirochetes in these mice were the same as the untreated mice, which means that the bacteria were there, And over time, they had the ability to resurge and to regrow. So this is a paper out of the Journal of Bacteriology looking at persistence. And this is basically just an outline of what you see with different types of bacterial growth. And so if you look at bacteria that are growing and you treat them with a drug, these are sensitive to the drug because they basically just stop growing at some point. However, persisters will, so this is an experiment where you would grow them in a liquid media, add the drug, and then plate them onto an agar plate. And persisters will grow after the antibiotic is removed, but they stay stagnant during the treatment with the drug. And then the non-culturable cells will remain alive, but they won't grow in the new media. So how do you resuscitate these cells? We've done some work in this area, and we've developed a new culture media that I'm on the verge of submitting for publication. It's been a long time coming, but we've shown much better recovery from non-human primates and from mice that are treated with antibiotics. So I'm just going to talk a little bit about the study that has been published for several years, but there are a couple of things that I want to highlight about it. So this is the design of the study. We had 10 animals, and we infected them by tick bite, and then we let them go for 4 months, and 5 were treated with doxycycline for 28 days, and 5 were untreated. And we performed a pharmacokinetic study prior to this to ensure that we were achieving the levels of antibiotic that we needed. And then we waited 3 months and performed xenodiagnosis, where we fed uninfected ticks on the rhesus macaques. And then we waited another 2 months, performed another xenodiagnosis, and eventually the animals went to necropsy after almost a year after the initial infection. And so a couple of lessons learned. First of all, only one of the animals developed what I would consider a bona fide erythema migrans lesion, whereas most of the animals developed this diffuse EM. And with respect to the antibody response, so what you're looking at here is a chart of the antibody levels over time to 5 different Borrelia antigens. And the light blue line here is C6. And so we've known for a long time that C6 declines, C6 antibody titers decline after treatment, and we saw this. But we also know that in the untreated, the C6 titers declined, for example, in these 2 animals that were untreated, but stayed high in these 2 animals. And this animal developed an antibody response that rapidly declined. This animal who was treated had no antibody responses that we could detect, period. And I initially thought this animal was uninfected, but I decided to continue the study. So what we know from this is that C6, which has been considered a possible test for cure, is unreliable because we know that C6 can decline in untreated individuals. So we're working on doing a better job of diagnosing Lyme disease as well, based on these longitudinal responses. And then when we looked at ticks that fed on animals that had been treated, what we call xenodiagnosis, what we found is that irrespective of treatment, we were able to recover spirochetes in those ticks. And these, for example, these are treated. This animal was untreated, and these are stains of the spirochetes from the tick midguts. And this one right here, clear positive. This was the animal that was completely seronegative. And then we have, we've done RT-PCR, which shows active transcription. And we picked up transcripts for OSP-C and OSP-A in the xenodiagnostic ticks from both untreated and treated animals, indicating that they are alive. And then when we looked at the histopathology, we found some very important things. These top panels just show that we use two different antibodies. The green is a polyclonal anti-Borrelia, and the red is an anti-OSP-A monoclonal, and this is the merge. Those are the controls. And what you can see here is some mild to moderate inflammation in the cerebrum here and in the brainstem. And this is the brain parenchyma. And what we found was a number of different spirochetes in the brains. And this is here and here. These are two different antibiotic treated animals. So we're definitely picking it up. We also found inflammation in the peripheral, in and around the peripheral nerves. We found spirochetes in those locations as well. This is the heart tissue from one animal. This is some interstitial inflammation and pericarditis right here. This is the layer outside the heart. And here is this beautiful stain with an OSP-A monoclonal antibody in the heart of that seronegative animal. And then oftentimes we would find this like spot staining or circular staining. And if you think about it, the way we're cutting these slides, we're cutting them at 16 microns. And so we're cutting these tissues at 16 microns. So if the spirochete is sitting flat, we'll catch the whole thing. But if it's sitting perpendicular to the cut, then we'll take a cross section. And this is what we see in some skeletal muscle tissue. We have staining with both the polyclonal and the monoclonal antibody. And when we did a rendition of the Z stack, so the Z plane, you can clearly see that this is a cross section of a spirochete that's dual stained in the skeletal muscle. So just looking at the histopathology report from all of the animals, you can see that these 5 are the untreated and these 5 down here are the treated. And there are 2 major differences that we noted. Ironically, for the treated animals, those were the only ones where we found spirochetes in the brain tissue or in the leptomeninges. And in the untreated animals, we saw more peripheral nerve involvement. So other than that, there's basically no difference between the pathology that we saw in treated animals versus untreated animals. So, you know, this argument over the viability of the spirochetes has been ongoing for a long time. Certain people will claim that they're not alive unless you can culture them. But what we did was we knew that there were a small number of spirochetes that we could find in ticks. And we did the RT-PCR, and we also injected those into severe combined immune deficient mice. And what I think is that, you know, we are kind of a dead-end host, right? We're not a reservoir host, we're an incidental host. And so it's hard for the spirochetes to jump from from us or from an incidental host back into a reservoir host. And so we really didn't get a productive infection of the skid mice. And we also, at this time, were unable to culture the necropsy tissues, whether from infected animals or uninfected animals. And so we decided to take pieces of the heart tissue from the infected animals and implant them into dialysis bags in the peritoneal cavity of rats. And this is something that's been done to grow Borrelia in a pure culture in a host adapted system, but we were doing it to see if we could get growth of the bacteria I'm calling this an in vivo culture system. And when we did that, the tissue came out necrotic. We let it go for about 20 days. But we did find spirochetes, and we did find transcript. And I sent this, what I did was I sent these samples to Emir Hadzik at UC Davis and blinded him to the identity of the samples and gave him controls. And he was able to find transcripts from 2 treated animals and 1 untreated animal. So clearly the spirochetes are alive and they're persisting in the tissues. So what we found in summary from this study was that erythema migrans was produced in only 1 of the 10 animals. All but 1 out of the animals seroconverted. We saw moderate pathology in both treated and untreated animals. We were able to find intact spirochetes by immunofluorescent staining of xenodiagnostic ticks and affected tissues. We were able to show persistence of live Borrelia by RT-PCR. We did send a bunch of tissues to directly to Dr. Hadzik, and he was unable to find transcripts, and I think that's because they're just so sparse. To find those samples in the brain, I think we went through 400 tissue slides. So they're rare, whether they're treated or untreated. They're hard to find in an incidental host, but they're there and they're alive. So we showed host-dependent signs of infection. Some animals were affected in the heart, some in the brains, some in the joints, very much like humans. We also observed persistent, intact, metabolically active Borrelia after antibiotic treatment. And what we think is that the histopathology findings support the idea that the chronic Lyme disease symptoms could be attributed to residual inflammation in and around the tissues that harbor a low burden of persistent host-adapted spirochetes and or residual antigen. And I will say that there are bystander effects too. There were certainly times when we found histopathology and we couldn't find spirochetes, and times when we found spirochetes and no histopathology. So whatever these bacteria are releasing, be it peptidoglycan or other biomolecules, they're still inducing the inflammatory response that leads to chronic disease. So this is just a paper that we published, a review showing all the different manifestations in non-human primates of persistence and inflammation. Okay, so now I'm going to move on to humans, because the criticism has always been, well, yes, you know, you can prove it in monkeys, but, you know, what about humans? There's no evidence in humans. So we did this, we received these samples from Brian Fallon and Andrew Dwork at Columbia University. And this case, I'm going to go through it in some detail, because I think it's really important. This was a 69-year-old woman who contracted Lyme disease at age 54. She had a well-documented EM rash. This is not a picture of her rash. This is just a picture. She had convalescent serologies that were positive on ELISA. She had a positive IgM and IgG on Western blot. And she was treated with doxycycline for 10 days, classic case. And this led to symptom resolution in the short term. However, 2 years later, she began to have sleep behavior disorder. She began to have cognitive problems, and this gradually worsened. She had neurocognitive testing. She had deficits in visuospatial skills, verbal skills. Clearly, she was going through a neurodegenerative process. The PET CT scans showed decreased perfusion on the right posterior parietal and temporal lobes. Parieto-temporal lobes are typically affected in dementia, especially Alzheimer's dementia. She had a PCR blood test for Bartonella, Babesia, and Borrelia that was negative. She was still positive for C6 by ELISA, and she was treated with ceftriaxone, IV ceftriaxone for 8 weeks, and this led to 60% improvement in cognition. And she was also given oral amoxicillin 3 times daily for 6 months after the intravenous ceftriaxone treatment. So she was treated extensively with antibiotics. However, her initial improvement was not sustained. And then she received more antibiotic therapy with minocycline with no clear benefit. She continued to be positive for Borrelia serology. She had a second brain MRI, which showed paraventricular and subcortical hyperintensities, possibly due to small vessel ischemia or demyelinating disorders like Lyme disease. She developed severe oral dystonia and died 15 years after the initial infection. So the diagnosis, the early and severe movement disorders, REM behavior disorder, paranoia, they were all consistent with a clinical diagnosis of dementia with Lewy bodies. And so we received tissues from her autopsy, and basically this is our process. We take the brain sections and we take an 80 micron slice, and then 10 micron slice, another 80, 5 more 10, another 80, 5 more 10. And we do this for 3 to 5 rounds. And from the large slice, we do a PCR. And if that's positive, then we do immunofluorescent staining in the tissues, the adjacent tissues for Borrelia burgdorferi. So when looking at the histopathology by the pathologist Dr. Dwork at Columbia, what he found was clearly consistent with dementia and Lewy body dementia. So these are Lewy bodies that were found in the substantia nigra with specific staining here and here. This is a Bielchowski stain here and here. These are Lewy bodies in this panel, and in this panel, amyloid plaques that are found in Alzheimer's dementia. And you can see multiple dark staining amyloid plaques in this patient's brain. And then, and that was in the amygdala, the amyloid staining. And we were able to pick up Borrelia in the amygdala in the pons of this patient by PCR. And this is a 16S23S internal ITS region. And the first panel here is the first amplification, and then the second panel is the nested PCR, where we amplify from that as well. And then when we did immunofluorescent staining, we were able to identify spirochetes in the spinal cord. This image is published, but this image is not published. As we continue to look in this this woman's brain tissue, we continued to find more spirochetes here. Here's another one that's dual staining. And so there's no doubt in my mind that this woman had a persistent infection with Borrelia burgdorferi in her brain. So our studies presented here and in animal models indicate that the Lyme disease spirochete can persist after conventional antibiotic treatment. And the case study presented here indicates that Borrelia can play a role in the development of dementia. We don't think that all cases of dementia are caused by Borrelia burgdorferi, of course, but we certainly think that Borrelia can induce the the series of events that lead to the development of dementia. For example, beta amyloid production is a defense mechanism for invading pathogens. And it's basically when it continues and isn't cleared, when the dementia, the signs of dementia begin. So what are we going to do about this problem? Many years ago, I paired up with Dr. Ying Zheng, who was at Johns Hopkins then, and he built his career in tuberculosis. And so we all probably know that the way you treat tuberculosis is not with one single antibiotic, because it's a slow-growing, persistent bacterial infection. We treat it with multiple different antibiotics. And so he began down this road of combination therapy. And after my non-human primate studies, I was thinking that we needed to do either pulse therapy or combination therapy to be able to eradicate the infection. And so these are some studies of different antibiotics that have been used in combination to kill Borrelia burgdorferi in vitro. And just a handful of studies have looked at the ability of different compounds to kill Borrelia in animal models. The first one here showed that log phase Borrelia could be eradicated by ceftriaxone, whereas the persistent infection caused by the microcolony forms could not be eradicated with doxycycline, ceftriaxone, or vancomycin, but they could be eradicated by the persister drug combination of daptomycin, doxycycline, and ceftriaxone. And this is another study out of the Rajadas lab showing that combination treatment of aslocillin with cefotaxime effectively eradicated the doxycycline tolerant Borrelia burgdorferi. So I think what's really important about these studies is considering Dr. Barthold and Hodzick's study, where they waited a year after infection to look for resurgence. So a lot of times you'll see mouse studies with antibiotics and for example, the hygromycin study, they waited 3 days after the end of antibiotic treatment and cultured ear tissue and said, oh, look, it worked, right? We don't take that approach. We wait for the infection to disseminate, we treat with the antibiotic, and then we let the infection go for a while before we try to culture or detect bacteria. And so we have 2 studies with combination antibiotic treatment, one supported by the Stephen and Alexandra Cohen Foundation in collaboration with Ying Zhang, and 2 another one funded by the Bay Area Lyme Foundation. And I'm going to talk about the results from the Bay Area Lyme Foundation funded study, because this has been posted in bioarchives. We're trying to get it published. And so you can see these 2 different study designs. Always we wait 2 months before we treat with antibiotics. And in one study, we waited 6 weeks and 10 to 11 months. And in the Bay Area Lyme study, we waited 2 months after the infection. And it turns out we can detect the spirochetes just fine after 2 months after the end of antibiotic treatment, but certainly not 3 days. So this study, we had a number of different antibiotics that we were testing: aslocillin, Bactrim, disulfiram. As you know, disulfiram is being used as a candidate antimicrobial. Aslocillin is also not, it's actually off the market, but it has a lot of potential, and we have a source to get the drug for testing in vitro. It's not used for humans. And then carbamycin, dapsone, rifampicin, cefotaxime, and loratadine, which is a histamine blocking. It's also known as Claritin. And the main idea I want to give you here is these are all the different mice that we tested. And this is in monotherapy. So they're all given one single drug for that period of 28 days. Some are oral gavage, some were IP, intraperitoneal, and one carbamycin was a subcutaneous. And interestingly, if you give a mouse oral gavage of antibiotics, you can be assured that they are receiving the antibiotics because they are unable to vomit. Rodents don't vomit. That's why rat poisoning works so well. And it's cruel. So, you know, when we looked, we have BSKH, which is the standard media, and then our modified BSK2, which is what I talked about before in terms of being able to better culture from different animals. And here you can see there was only one drug that was statistically significant in terms of reducing infection, and that was cefotaxime. And we still had one of 5 animals that was culture positive. However, when you treat with combinations of antibiotics, we saw 0 in all of these different combinations, 0 of 5 mice in each group that were culture positive. And so, you know, even doxycycline plus ceftriaxone is a lot better than either one of them individually. Dapsone we found was pretty effective, was highly effective in combination with multiple drugs. Loratadine didn't add anything because cefotaxime and doxycycline on its own was effective. Cefotaxime and carbamycin was effective. So there are a lot of different combinations that that have the potential to eradicate the infection. So what we've learned is that doxycycline and cefuroxime in monotherapy do not clear the established infection. In general, single drugs were not capable of clearing the infection, yet there are certain drugs that show promise. And we do have a study that we advanced to non-human primates. And this was from the Cohen, Stephen and Alexander Cohen Foundation funded study. And that's very labor intensive because you have to do pharmacokinetic analyses of all the drugs that you're testing. But it's much more like humans. We do infection by tick. The dissemination kinetics are very much like humans. We wait 3 months, treat for 30 days, and then wait 5 months to look for persistent infection. And I don't have data to show you for this, but I can tell you some general lessons that we've learned from these studies. The first is that ineffective treatment may result in very poor outcomes. And I think, you know, the fact that we were able to see spirochetes in the brains of doxycycline, 28-day doxycycline treated monkeys and not the untreated is really concerning. So I think it's really important if you treat early with microbiostatic antibiotics, you have much better outcomes, but treating disseminated infection with microbiostatic antibiotics can result in very poor outcomes. And secondly, we've learned that it's much easier to cure mice than to cure primates. And so I'll just leave you with that. We continue to work very hard to find effective treatments. We also have a collaboration with Duke University to look at identifying small molecules. Duke basically identifies these small molecule inhibitors of Borrelia and Bartonella. And then we have the model systems to test them. And we're currently collaborating with them on testing small molecules that are candidates for killing Bartonella in our model system, which is a immunodeficient mouse. And so with that, I'd like to end my talk. I could not do any of this work without a fantastic group of people that work in my lab, students, technicians. They're all very talented. A lot of the pathology was done by Dr. Crossland, who is an anatomic pathologist, a veterinary anatomic pathologist, who's now at Boston University. And my funding from the Stephen and Alexander Cohen Foundation, Bay Area Lyme Foundation, Global Lyme Alliance, and Focus on Lyme. So with that, I'll take any questions.
So thank you, Monica, for the excellent talk. There is one question here. Do you think there could be possible perspectives to resuscitate dormant bacteria in order to make them more susceptible to usual antibiotics? Do you see that question?
So are there ways that we can, with medicinal chemistry, induce bacteria out of the dormant state?
Mm-hmm. Right, that's the question that's been
Yes,
asked.
it's my question.
Yes.
And
That's a
congratulations.
really, that's a really good question. And I don't know, but we did this study and I'm just super excited about it because what we did was we have mice that are infected with Borrelia burgdorferi that produces a luciferase. And so you can do live in vivo imaging of the bacteria in the mouse. And what we did was we infected the mouse and we let it go for 28 days. And we saw the bacteria in the joints, basically, and in and around the mouth, which is common with when you do imaging of mice. And then when we placed ticks on the mouse, all of a sudden the bacteria started to proliferate and spread throughout the body. I thought that the bacteria would go to where the ticks were feeding, but I think they just re-enter the circulation. And so now we're looking at tick salivary proteins for the ability of them to change the phenotype of the bacteria so that they re-enter the circulation, enter that dissemination phenotype. And if we can do that, then I think we have not only increased our potential for this as a diagnostic tool, but also as a therapeutic tool. It's kind of pie in the sky, but tick saliva, stay tuned.
Okay. And then
Thank you very much.
just one question from the audience, and Harold, your comment?
Can you hear me, Monica? Can you hear me?
Yes.
Your data on the doxycycline was quite worrying that you found it drove the brain. Any ideas why that might be the case?
So just to repeat the question so everybody knows, doxycycline doesn't cure, and if anything, in the treated patients, that there was more evidence of brain involvement of spirochetes when doxycycline was used. That was the question, and why is that? And
Yeah, we don't know specifically, but something that we've been curious about is that we see re-expression of outer surface protein A, which, the dogma is that OSPE is downregulated as soon as the spirochetes get into the host from the tick. But at some point it's upregulated again. Like I said, we saw it in the heart. And I almost think this is a marker of dormancy, because we did a study where we treated Borrelia with doxycycline in vitro and looked at the genes that were upregulated, and OSPE was one of them. And we also see upregulation of OSPE when you look at spirochetes in the brains of non-human primates. So I think doxycycline might be inducing a phenotype that allows for more extravasation into the brain. And we could also argue that, this whole gut-brain axis where the antibiotic treatment could actually affect the permeability of the blood-brain barrier. So there's a lot to explore there.
Just two more questions, I think, from people in the online. Just one question is, what percentage of your research funding is private versus government funding right now? Just a ballpark figure.
Probably 75 to 80% is private.
Okay, and then there was just Amir Kadir has his hand raised. Do you have a question you want to ask?
I was just curious, based on the do you hear me?
Yes.
Yeah, based on the latest study that appeared in emerging infectious disease by the team of Frank Sterl in Europe about the circulating interferon alpha among neuroborreliosis, post-treatment neuroborreliosis. So I was asking if the anti-interferon alpha activity of doxycycline might be a promoting factor for lowering immune response, which could in turn favor brain infection. Could it be a pathway?
Yeah, that's a very good question. And I think those, those studies from the Sterl lab are really important in looking at how does Borrelia burgdorferi induce this inflammatory response that persists. And yeah, I think the jury's still out, but that's very important.